Welcome to today's USP PharmaTech webinar, Modern Approaches to mRNA Manufacturing. Thank you for taking time out of your busy schedule to join us. Next, please. Each registration is unique and grants the right for the individual registrants only to view this webinar. Sharing this webinar with unregistered viewers, either during or after its performance, violates PharmaTech Associates' copyright and is prohibited. Next, please. If you have not already done so, we ask that you take some time to read the PharmaTech Associates course disclaimer. You can download this presentation, including the disclaimer, from your USB education account. Please follow the instructions in your confirmation email to download the presentation. Next, please. We have two presenters today's webinar, Apur Puri, Principal Engineer at PharmaTech Associates, and Adam Lampert, Practice Lead of PharmaTech Associates. Presenting today, first is Apur. Apur, go ahead.
Hello, everyone. Thanks for joining us today for this Modern Approaches to mRNA Manufacturing seminar. Really, today we're going to be kind of delving a little bit deeper into some areas of development, especially manufacturing innovation, that's going to kind of enable the next generation of mRNA engineering and production. So to that end, we really will be going into roughly four areas today. One, we'll just kind of do a brief overview of mRNA production and mechanism of action. And it's just to briefly orient you and kind of give you some refresher or review, and then really kind of looking at current state and challenges with this mRNA modality. Ultimately, many of the tools utilized even currently are from legacy processes. And then given that emergency use authorization was provided for the COVID-19 vaccine, really the technology grew much faster than the supply chain's ability to support it. And really there's a ton of innovation on the horizon. It's almost impossible to really cover it. We'll be looking at some exciting stuff, albeit a little bit of a biased opinion, but at least some of these technologies and innovations that are being explored. These include microfluidic technologies, potential ideas for continuous manufacturing, and then ultimately looking at enzyme and protein engineering. And then really kind of what's needed for the future to hopefully encourage quicker technology adoption. So let's get into the mRNA production and really mechanism of action. Here's kind of more of a high-level layout of current process flow for mRNA production. It really begins with a templated plasmid DNA strand that is linearized, and then through enzyme transcription, you generate the mRNA template. And then once that's generated, you really want to be able to digest any plasmid DNA that's left in there, and then begins kind of your downstream purification. And this is typically a multi-step operation. Pretty much every company with an approved mRNA therapeutic, or at least in development, they keep their downstream kind of a little bit proprietary, but it can really consist of multiple chromatographic steps, and then to render really stability to the mRNA so it's not digested or degraded immediately following kind of an in vivo delivery. You usually enzymatically cap the mRNA on the 5' end to be able to render it a bit more stable. And then really ultimately, since we do have to deliver this to a patient, there's multiple formulation steps that not only render this further more stable, but ultimately they also provide the ability to deliver the mRNA to the targeted tissues or whatever. And then ultimately, it's finally sterile filtered, goes through a capping and sealing process, and then ultimately your labeling and packaging. So let's talk about the mechanism of action a little bit first, really quickly. Once you have your in vitro transcribed mRNA and it's loaded into either cationic lipid nanoparticles or polymeric nanoparticles, the way it's really uptaken into the cells is via endocytosis. And then once it goes through endosomal escape, the mRNA is now open for translation via ribosome. And then once that protein is displayed, either it can get degraded within the cell itself, but the way you usually have this presented on the surface of cells is via this major histocompatibility complex. And this is really kind of the way this MHC class 1 peptide or class 2 peptides, once they're presented on the cells, CD4 positive and CD8 positive cells can really act on that. And then this becomes kind of the antigen that's recognized for a further immune response. And now to get into a little bit more around the process, just to kind of give us more of a foundation of kind of the later subjects in the talk. It really first begins with this in vitro transcription. So as I'd mentioned, you really need a linearized plasmid DNA template that a synthetic RNA polymerase can translate. And then this begins kind of your mRNA transcription process, and then ultimately produces the therapeutic mRNA for downstream capping and purification use. And then one thing I want to draw your attention to is really this will kind of provide impetus for enzyme engineering later on, is really this kind of steps of initiation, chain elongation, and ultimately termination once you encounter the stop code on the linearized pDNA. And one thing that kind of happens with basically any T7 or any RNA polymerases that are being used, as it first binds to the pDNA, there's these short RNAs or abortive transcripts that are kind of produced. The mechanism for this is yet unknown, but the hypothesis is that as the RNA polymerase binds to the DNA, it goes through several conformational changes, and until it reaches some sort of thermodynamically stable conformation, it will produce these short abortive transcripts. But once it's fully bound and into its more thermodynamically stable conformation with linearized pDNA, then the elongation step begins and you start to get your therapeutic mRNA. One thing to note that these short RNA transcripts can kind of loop back on themselves and form double-stranded RNAs. Same thing with longer transcripts. And then what ends up happening, unfortunately, is that if these end up in your final product, these can be immunostimulatory and can produce unwanted immune responses in patients.
So, no, that's good. No, thanks for doing this. This is great. Well, when you point back to the loop back and over on the bottom right-hand side of the slide, you have the three-dimensional structure, right? I mean, a secondary structure of the mRNA. Can you talk a little bit about what role do the secondary structures play? Do they confer stability? And is there a way that people are looking at to exploit them for purification?
Sure, I think that's a great question. Secondary structures, in fact, do render some level of stability to the mRNAs. One thing to also keep in mind is that secondary structures will actually also determine how well they bind to certain ribosomes. So ultimately, this has implications to the biological stability of the molecule. Now, as far as potential exploitation of these secondary structures, there's now, I think as of the last 18 months or so, people are looking at ways to exploit these secondary structures for purification applications. This has never really been explored before. but certain hypotheses seem to state that potential ultrafiltration or even chromatographic approaches could benefit from better characterization of these secondary structures.
Great, thank you.
And really, once your mRNA transcript is produced, there are a couple ways to enzymatically cap the mRNA to render it further more stable. So, and the reason you want to do that is because if you really don't have this 5' methylguanosine here, this ends up rendering the mRNA open to degradation. So once you deliver this, if this is not present, not only does it degrade the mRNA, but obviously reduces your therapeutic potential. So the two ways to do it really is post-transcriptional. So once the mRNA has been transcribed by the T7 RNA polymerase, you use additional engineered enzymes, either Faustovirus or vaccinia capping enzymes, to be able to add this additional phosphate conjugated to methylguanosine. And then if you were to look to reduce costs, you can certainly do it in a co-transcriptional manner with these kind of anti-reverse cap analogs or engineered clean cap reagents that are available. And these are slightly different as there's additional 3' O methylation that is added. But in the case of clean cap reagents, ultimately this ends up the same. And what I mean by co-transcriptional is that really this you can add these enzymes into the same mixture, into the same in vitro transcription mixture. And once you have your capped mRNA, really now you have your kind of drug substance, therapeutic substance of interest. But unfortunately, even post-DNase digestion, you still have quite a few double-stranded RNA transcripts, potentially other impurities, free nucleotides. So the first step is really to try and retain the mRNA that you have. And so really, there's two modes of filtration uh, that are frequently employed. You guys are probably familiar with dead-end filtration, which is really running the flow kind of perpendicular to your filtration membrane. But as you can likely guess, as you filter more and more substances over time, this membrane can certainly become fouled. And so it is trapping, it is working the way you want it to, but it's going to require a little bit more push, a little bit more, higher flow rate, and certainly this back pressure is going to generate some shear forces. And if you know anything about nucleic acids, is that they don't behave really well under shear forces. So we want to entirely avoid that. One kind of a great way to do so is instead of having your feed flow perpendicular to the membrane, have it flow parallel to the membrane. So you avoid these fouling issues. You don't have to generate higher and higher pressures to combat the back pressure from the membrane. And really, a consistent transmembrane pressure should allow you to retain your mRNA on the flow side of the membrane. And then, really, the stuff that falls through in the permeate are your short abortive transcripts, anything that's really smaller than the capacity of the membrane. So typically 100 kilodaltons is like the molecular weight cutoff for membranes that are used within the process. And the nice thing is that tangential flow filtration is really kind of a legacy tool from monoclonal antibody purification and really other purification processes that have been used in the past for biologics. And there are really fully functioning automated platforms available. These systems are also easy to customize, and you can really spec a custom system. And then ultimately, the other thing is we want to be able to wash our product. So this unit operation really does provide nice buffer exchange capabilities to facilitate downstream chromatographic operations. So let's get into some downstream chromatography. One thing I want to draw your attention to is that chromatographic materials have changed a lot over the years, really from kind of first generation membrane materials to kind of more porous materials to ultimately bead supports. And really we're on this fourth generation here of monolithic, almost sponge-like material that really kind of combines all three of these previous generations modalities. And then I show a sample, chromatographic graph here. So really, once you the operations are essentially composed of loading your mRNA product onto the column. So that's typically what's done with the buffer of choice that you've already kind of exchanged during your ultrafiltration step or your microfiltration step. And then as you go through this process, what you're doing is you're loading the product on and then changing the buffer composition in such a way that your conductivity goes down. So you lower really the ionic strength of the buffer. And then as you do so, the mRNA molecules that are bound to the chromatographic supports as part of the loading process are then subsequently eluted. And the way you can kind of see this on a chromatogram is by measuring UV absorbance units at the 260 nanometer range. So you'll see a peak correlated with your eluted mRNA material.
Okay. Yeah. So I guess, one of the questions here that I have, Apoorv, is that, for the fourth generation monolith, what are advantages and what are the bottlenecks? Because I'm kind of, the downstream chromatography, you mentioned limitations due to shear, right? And things along those lines. So if you could speak a little bit to these advantages that the monoliths give and where the bottlenecks, does it relieve the bottleneck or does it, at least partially remove it?
So prior generations, really, the one thing, beads and kind of porous, solid chromatographic supports, really the thing they suffer from is similar to kind of the back pressure situation I alluded to on the ultrafiltration side, but really for different reasons. And so as you load up the column really, these solid supports, you really don't have very high throughput, high flow rates. So chromatographic operations do take some amount of time. The unit operation time is on the order of hours, and so as you get more and more upstream product, the fact that you have to equilibrate the column, the fact that you have to load the column, and then now you elute the material of interest, all of that takes some amount of time. And then since the back pressure situation was very much present with kind of porous and bead-like supports, the monolith supports, since they're a little bit more like sponge-like structures, they do provide higher convective flow because they're composed of these microchannels that allow you to flow your product through. And there's really a lot less back pressure, a lot less, the application is certainly more low shear. And then as it provides those convective channels, it also provides larger surface areas for more product to be bound. And so there's a slight increase in kind of binding capacity with lower pressure drops, lower back pressure at higher flow rates. So the technology is changing, but I would say this is not the most high throughput operation, even with kind of fourth gen monolith supports.
Okay. No,
And
that's good.
then as we get to a pure and purer product, now that we have our mRNA at the desired purity, the challenge now is to conjugate this into something that's going to be able to be delivered to your target tissues of choice or intravenously or whatever. And really kind of the way folks do this is via conjugation to polycationic lipids. So since the mRNA is negatively charged, once you mix it with these kind of positively charged liposomes, what ends up happening is as you mix these two solutions, one is combined with your stabilizing buffer and your mRNA, The other is kind of a composition of a solvent like ethanol and your cationic lipids. As you mix them in microfluidic channels and your ethanol concentration goes down, these mRNAs start to form these micellar structures that are, that contain that basically are of nanoparticle size and contain your therapeutic mRNA. And this increase in polarity during mixing really causes formation of size-limited, thermodynamically stable particles. And the other thing is that pretty much in every clinical study reported, at least using these liposomes, they offer nearly 100 to 1,000 times better delivery efficiency than potentially current polymeric solutions or otherwise.
Okay. So now from looking at microfluidics, is that a potential downstream bottleneck as well?
It certainly can be, mostly because you have to now take a bulk solution and run it through microchannels. But typically, the way folks have been kind of either combating this is via just addition of more parallel channels. So you just have the ability to have higher throughput. Or some form of different customized microfluidization equipment that is able to mix a large solution while still passing the solution through what looks like a microfluidic channel, so you can start to get these conjugation complexes starting to be formed.
Yeah, and then a little bit, if you could talk a little bit about the stability issues that people need to be aware of as well, and critical process parameters, and how are they going to impact the process?
Certainly, and I think the thing to really consider is that we really want consistent sizes, so you can deliver therapeutic mRNA payloads that are consistent between vial to vial, between lot to lot, and the way to really control that is, via flow rate, via proper mixing, via microfluidic channel design. And so you can get low polydispersity, so you don't get nanoparticles that some are 10 nanometers in range, others are hundreds of nanometers in range. You really start to get these much tighter size distributions. So best way to really control that is kind of via mixing. And then mRNA is notoriously unstable. So typically temperature control is pretty important to this process.
All right.
And we'll kind of get into at least the current state and challenges, and into the later half of the talk. Really the things maybe I've already alluded to are kind of this, manufacturing systems and really tools and technologies deployed from previous legacy manufacturing platforms, really used for biologics at the end of the day. And then since the technology developed so quickly under emergency use authorization, there's a little bit of uncertainty around what level is really required for raw materials, especially templated DNA, synthetic DNases, and enzymes. We really need to know what level of fidelity is required for the mRNA, even though the things that are approved on the market are certainly therapeutic, and that has unequivocally been proven at this point. But still, for other modalities, for mRNAs with less stable secondary structures, can the same polymerase provide the same level of fidelity for other applications? That's really unknown. And then reagents have been kind of the supply chain is not certainly robust enough. And then you also suffer from kind of the classic downstream bottlenecks of downstream purification, where binding capacities and throughput are still somewhat limited if you're using older generations of chromatographic technology. And then even though standardized platforms are starting to become available, there's still no kind of standardized in vitro transcription reaction or capping or end-to-end platforms that are available for some of the upstream processing. So let's talk a little bit about more exciting things now that we've kind of at least completed the review of of the process and kind of gotten a little bit of background to maybe think a little bit more innovatively around, what can we do to kind of speed up this process? What can we do to make it better? What can we do to get higher quality product at the end? And so we've kind of talked about a lack of kind of engineered, tailored reagents. So we'll kind of get into that a little bit. And then given that this is an enzymatic process, it is really open to continuous processing. So you don't suffer from having cell systems that have pretty finicky growth profiles, and then you don't have to perform these extensive and kind of tedious lysis processes that can be hard to control if you don't have good scale-up approaches. And so we're kind of at least free from those issues. And then if we were to integrate this via kind of online real-time process analytical technology via HPLC UV and some other technologies, we really could potentially, provide real-time analytics of nucleotide consumption, how your reaction to produce your mRNA is progressing, and then ultimately, do the same thing with downstream operations. And then we'll talk a little bit about platform systems and automation. Please keep in mind that this is not an endorsement of any single one technology. We're certainly just discussing what's starting to become available so that you guys are familiar with companies kind of making the efforts towards standardizing platforms for nanoparticle production and kind of real-time analytics with some chromatographic processes. And then really looking at potential multimodal and more so, I would say, monolithic chromatographic operations integrated with kind of process analytical technology. So we'll talk about, I think, what is the most exciting thing for me is really rational design and protein engineering. So what you're really looking at here on the left side is this T7 RNA polymerase, kind of this protein map. And so at the top, you see this kind of DNA binding to this T7 RNA polymerase. And ultimately, researchers at Moderna, this is kind of their work published, I believe, earlier this year. So very modern, very hip, very cutting edge science, really looking at the amino acid structures and then the corresponding void volumes that are shown here. So void volumes are really the empty space. And so if their hypothesis is essentially that if they can potentially engineer these void volumes by engineering the amino acid residues that are here, they're thinking that they can provide better binding of the plasmid DNA and then ultimately less potential for these impurities that are, short RNAs or smaller double-stranded RNAs that can be formed. And so on the right side here, you really see kind of a radioactive gel that shows, the full-length RNA produced by the wild-type enzyme, as well as this engineered enzyme. So this, all this really means is at what amino acid residue, what residue was changed exactly which one. And what you'll see is that in their engineering efforts, they were able to show that both loop-back double-stranded RNAs and smaller double-stranded RNAs can be almost essentially eliminated from this in vitro transcription reaction via engineering of this T7 RNA polymerase. So I think this is pretty exciting stuff. And then the additional thing that the scientists also are potentially looking at for the next generation of engineering of this molecule is to really look at the N-terminal domain. So these residues are really on the C-terminal side of the RNA polymerase, but on the N-terminal side, where additional binding occurs, it's possible that that is really how you'd end up reducing the short RNAs as well. And then really getting into potential other continuous ideas. So I think if The in vitro transcription upstream reaction can be made continuous through either kind of a continuous stirred tank reactor design, where reagents are essentially continuously fed and then taken off the reactor. Uh, Being able to perform some sort of continuous chromatography steps downstream can be pretty valuable. And so some ideas that I think can certainly be tried are to essentially run two chromatography columns almost in a counter current fashion. So what you'd end up doing is watching and equilibrating column one. And then as you complete this wash and equilibrate, you start to load up your product. And while you're loading and eluting the product on column one, you're getting a second column ready to continue to process the upstream pool. Then you switch to the second column, and as you're loading column two and equilibrating or eluting, then you're washing and equilibrating column one again to be able to load additional product on there. And so in this kind of manner, you're basically serially loading one column while getting the other one ready, and then as you elute the first column, you start to load product onto the other one. And really, I think this is one way to kind of think a little bit more, not necessarily outside the box, but a little bit more creatively about how you can start to make these processes at least somewhat semi-continuous. So not only do you get higher throughput, but you kind of potentially deal with some of the chromatography issues that you would typically encounter. And then really additional topics, I personally find this very interesting as well, where, we've kind of explored these cationic lipids, but what about, exosomal structures? So we already have exosomes kind of in vivo that really perform the packaging job across, our bodies really well. And so being able to exploit these are kind of natural vehicles. They're not going to be immunogenic. And ultimately, they are controlled to kind of a 50 to 150 nanometer size. Same thing with chitosan nanoparticles. And then being able to load, via different ways. So currently, encapsulation is kind of the major way, but if you could polyplex or adsorb these onto inorganic materials, this can certainly provide a much higher benefit. And then one thing to note is, via exosome engineering and kind of other methods utilizing, really native organisms biology, you can potentially even target specific tissues. And so no longer do you have this potential for maybe off-target introduction of your mRNA. You're really able to kind of engineer almost like a homing missile with your mRNA payload. And although inorganic materials haven't really been explored for this application, it is important to know that inorganic materials, especially things like gold, are very inert, and so this certainly offers a potential for uh, you know uh, adsorbing certain molecules onto the surface of these, conjugating them, and really being able to provide, I think, superior circulation with continued low toxicity, because certain inorganic materials are already used for imaging purposes, and so this is really not outside of the realm of exploration. So these are starting to engage scientific communities across the world, and especially if we can get targeted deliveries of mRNA, I think we're opening a whole new, area of scientific innovation that I think is quite exciting.
Good. So with regard to the customized delivery systems, can you talk a little bit about what some of the reasoning is behind using them? What are the risks to patients with regard to safety? And then with targeted, you know, are there off-target effects? And how do these play a role in selection of this in the overall product, stability and shelf life?
Sure. So cationic lipids, I think, are limited in the sense that they only offer, um, at least under the current modalities, kind of intramuscular delivery. And so one way, and I've kind of alluded to maybe conferring more tissue specificity at the end of the day, kind of utilizing maybe more engineered particles. And so that certainly is, I think, one advantage of kind of using different delivery systems, because I think it's if I've learned anything over the years, is there is no one solution that exists in kind of any scenario. So uh, really being able to engineering something specific for, let's say, a liver or spleen, I think that's a pretty big deal. And then one other thing that's been historically just hard to do is delivery across the blood-brain barrier. So being able to have something that can pass through the blood-brain barrier, offer that kind of level of specificity, but at the same time not being immunogenic, I think is quite the advantage that just hasn't been well explored. And then as far as really off-target effects, I mean, those are certainly possible now. And so I think from a motivation perspective, really the off-target effects are the reason to develop some sort of customized delivery system so you can target certain tissues much more efficiently, get higher perhaps transfection efficiency is not the right term, but really getting much higher expression in those target tissues. And then from a toxicity perspective, I mean, really as long as the engineered material is compatible with what it's being delivered to, or in the case of like inorganic materials being completely inert, I think these still offer far less toxicity risks than kind of uh, traditional, delivery methods, either via kind of injection formulation or otherwise.
Okay.
And one other, I would say, kind of synthetic solution that's being explored are really these polymeric micelles. So one potential is this kind of ether backbone that really offers more of a non-polar end, and then these NH2 amine sides that really offer the kind of the polar end of the molecule. So this negatively charged mRNA can conjugate pretty nicely to the polar end. And then the one big thing with this is really you can control the size, and self-assembly just happens. So you're not limited by microfluid, the need for kind of some sort of microfluidic machine or microfluidization platform. These just kind of self-assemble. And then if you engineer the size kind of very intently and very rationally, you can also control the size to very narrow overall dispersion. So size can be controlled a lot better through this kind of a design. And one additional advantage is that really these polymers are all biodegradable. So as they degrade, they release the mRNA therapeutic payload. And then since they're thermodynamically limited to a certain size, one other advantage is that you're going to be able to not only get low polydispersity, but you get consistent loading of mRNA, because really it's almost like a stoichiometric conjugation. So every chain of the polymer is conjugated to a certain amount of mRNA. So this offers, I think, much better control and way higher potential for higher throughput, so that kind of some of these downstream bottlenecks that we've been talking about are at least somewhat relieved.
That's good.
So let's go into some microfluidic system design. We've kind of talked about some, kind of the importance of these in being able to generate nanoparticles of somewhat of a uniform size, and then really get them conjugated in a way that allows you to deliver your mRNA to the target tissues or whatever you're end goal is. And really currently lithography, micromilling, laser ablation is used, and so the channel designs are very precise. The standard herringbone mixer I show here in the middle is really kind of the one that's used most frequently. But obviously, there's multiple designs depending on how much, what different reagents you're mixing, what level of mixing you want. And then really the control of these channels offers kind of control over particle sizes and um, can really give you a lot more control over um, just your particle quality. So certainly, I think baffle types and bifurcation ones have been less explored. But if these could offer low polydispersity, better conjugation and better loading of mRNA, better encapsulation efficiency, these are certainly worth exploring. Although I will say one word of caution is that, the standard herringbone mixer design is quite good. It does offer, in most cases, higher than 98% encapsulation efficiency. But, if we talk about how much mRNA is really required to have a therapeutic benefit, even losing 2% mRNA due to this process is likely not favorable. In most conditions, I think we want a high level of conjugation. We don't want to be losing mRNA, mostly because if you lose that 2%, that's potentially hundreds, if not thousands of doses that are going to waste because you're not loading the full amount into your cationic lipid particle.
Yeah. So yeah, so there's different kinds of control. I mean, so would you suggest trying different, you know, channel design out as part of your process development?
Certainly, I think if you're not following your traditional cationic lipid process, I think different designs might be almost required to be explored because let's say you are using, different polymeric particle that maybe doesn't offer good control under aqueous mixing conditions. So if you were to design a microfluidic channel that offers specific fine tuning and mixing for that polymeric particle, then I think it's definitely worth exploring, mostly because I think controlling the optimum flow rates and mixing speeds and mixing rates via the proper channel design and proper flow rates can offer you, I think, much better control. So I don't think the standard herringbone mixer design works in every situation. I think certainly this is great for cationic lipids as it's been shown, but ultimately I think there is some development work if you're that's going to be required that if you're really looking at different ways or different conjugation materials to conjugate the mRNA to.
Okay.
And then really the last portion of our talk is kind of looking at platform systems, how these platform systems are going to offer hopefully better control. And then given that the current process could be potentially manual, being, having some option for automation can certainly give you much more consistent product through clinical. And then given that these are kind of small, parallelized, uh, these are kind of small, almost scale out type of systems. Really, if you need to increase capacity, you can just put additional systems side by side. So, and the other advantage of really some of these platform systems is that they offer a very controlled environment. They offer adaptation to most workflows. So anything that's currently looking at mRNA production, it really stands to benefit from a system like this because you can offer consistency from the preclinical stage all the way to potentially commercialization stage. And then you don't run into these situations where you potentially have comparability issues or you have to show kind of a lot more level of characterization and development work to be able to say that you can properly scale up your process and you are getting a comparable product at the end of the day that has the same, functional same function and same therapeutic benefits. So the preclinical development is kind of already at the final scale. All you'd have to do is kind of scale these systems out. So I think this is really a nice system to be able to adapt to really any kind of current workflow. Obviously, I think it would be a little bit tougher if you had a need for custom systems, either upstream or downstream. But for most, I think process workflows, this is certainly something that can be explored. And then additionally, we had talked about process analytical technologies to give you better insights into how your process is progressing. So Sartorius has really come up with this PATFIX system that not only combines kind of this in vitro transcription reactor, but it has real-time analytics on the nucleotides being consumed. So here you'll kind of see the nucleotide consumption in shown in millimolar, and then on the other side, kind of measuring mRNA concentration. And so you can really monitor your IVT reaction real time by looking at how your mRNA is progressing throughout. And this maybe potentially even gives you greater control of your process. So just because I think most mRNA processes take around, IVT reactions take around 4 to 5 hours, doesn't mean every single process takes that amount. So really understanding to what level your reaction has proceeded to completion. And in some cases, you might be limited by the concentration of your nucleotides. So if you do need to feed more nucleotides to kind of speed up the reaction, this will certainly give you the insight and the analytics to be able to do so. And then one additional thing to really think about is we talked about, combining PAT with also chromatographic systems. So if you could combine your chromatographic system with real-time analytics on your eluent that's coming out of your system, being able to analyze the amount of free mRNA, free pDNA, enclosed mRNA, enclosed pDNA, and the concentration of your lipid nanoparticles really gives you much better real-time analytical control. So you don't have this lag between your process ending, sampling, and then sending your samples out for testing. Being able to have real-time analytics, I think, for me, is really the next generation and kind of the future state of this. I think less and less you should be seeing kind of these traditional processes where you run the process and then you really have this lag time between when your process is complete and then when you actually get information on how your downstream unit operation went.
Okay. Yeah, I have a question here, but I think we're running low on time, and there's a number of questions in the chat. So I think
Sure, I can certainly move forward. And then really one additional technology I'd like to draw your attention to, we had been talking about nanoparticle size control, as well as being able to have some sort of a GMP system, some sort of a platform system to be able to take your therapy commercial. Really, Precision NextGen is kind of offering these scalable, modular, configurable systems for microfluidic processes. And again, much like the Quantum system, it really is a generic process, adaptable to most workflows utilizing cationic lipids. And then one additional thing I'd like to draw your attention to is really, if you look at kind of old microfluidic processes and the current next-gen process, the polydispersity, really the range of particle sizes, is much lower than your traditional process. So not only are you getting consistent cationic lipid and mRNA nanoparticle sizes, this should offer theoretically better loading as well. So if you're getting consistent loading, you're getting a consistent dose, and you're getting pretty high encapsulation efficiencies on the order of 90 to 95%, which I think is quite great. And then really here, it compares the Ignite to the Blaze to the full GMP system. So one additional benefit of this is, scalability from maybe your preclinical to clinical to commercial platform, and not having somewhat kind of comparability issues essentially, right? So, because if you had low polydispersity in one system and very high on your GMP system, can you really guarantee you're offering the same therapeutic dose across systems? So, one additional thing to consider, and I really like this because I think it offers drug sponsors a way to really, remove headaches of going and scaling up a process, because they've shown the level of similarity each of their systems have, and the level of high encapsulation efficiency that you would expect from a GMP ready system.
Okay. Now, do they have an idea of the ideal size for consistent endocytosis and delivery?
I mean, I think that's a good question. I would say, in my limited experience, that I think it depends to a certain level on the tissue or kind of the target you're delivering to. I would be shocked to find that, delivering to an organ, it requires the same particle sizes as maybe an intramuscular injection. But certainly that's something that's the current area of inquiry. I think that needs to be better explored. 'Cause I think it would depend on the materials. It would depend on the therapeutic payload. I think there are a bunch of variables to that puzzle picture.
Oh, thank you.
And then really to wrap up our talk, we kind of talked about the existence of legacy technology within the process, really how much of the mRNA process really still used is downstream operations that were really developed and fine-tuned for kind of other biologic products. And then given that certain challenges still remain around reagents, proper enzyme design, and then really what raw material quality contributes to a nice final product quality, that is still that still remains unknown. But and process scale up can be poor, but as we showed today, there are certainly platform systems now available that should lead to at least mitigation of some of these issues. And that kind of leads into our ongoing technologies that we really talked about, everything from highly engineered proteins to better kind of these multi-step continuous type of chromatographic approaches, using fourth-gen materials that offer a little bit better throughput, a little bit higher binding capacity, and then really kind of these platform systems. So I think moving forward, obviously still some challenges remaining. We're really hoping for faster, better engineered enzymes that can lower the duration uh, of the IVT reaction, hopefully better downstream separation processes, and more flow through type processes that we can get that maybe don't rely on the microfluidic channel design. I think certainly can be explored. And then I think there's no reason to discount inorganic materials as possible delivery vehicles. So those can certainly be areas of future exploration. So with that, I'd like to at least thank you for your time. I think there are some questions that are open. Christy, how would you like me to proceed?
I'll go ahead. This
Okay,
is Sylvia.
we got
Yeah,
that.
go ahead.
All right,
Yep.
we got it.
So thank you both for your knowledge and instruction this afternoon. Using the Conferences.io software, please submit your questions through the social Q&A interface. You will also have the option to upvote a question by clicking the arrow next to the question. All right.
It looks like we have
You can go ahead and
quite a
respond.
few. The one on top, I guess, is the number of votes that it has, the number of people that have clicked it.
Yep,
Um,
looks like it.
do
So
you
I think
want?
for the first question, I mean, so options and strategies for us to be able to analytically differentiate double-stranded RNA as a process-related impurity versus secondary structures. So I mean, one way to certainly do it is, you run it through kind of an online HPLC process. I think to be able to differentiate that in your IVT mixture can certainly be possible. I don't know if that's really been explored, to be honest with you. So,
Yeah.
if at all possible, I'd like to be able to answer that offline after potentially looking at a few sources. So, in terms of being able to comment on the exact options or strategies, unfortunately, I don't know if I can actually provide the answer that you're looking for.
Yeah, with
Um,
regard to the analytical stuff, I mean, I can speak a little bit to that, but
sure, yeah, go ahead.
you know, there's a lot of different techniques for separating molecules. The the binding, right? You know, I mean, there's size exclusion chromatography that's used
Yep,
as a technique, right? People are using gel electrophoresis, you know, but you can also look at DSC, right? Thermal denaturation studies for, you know, double-stranded DNA and RNA. You can look at mass spectrometry. An MR is also another powerful tool. CD spectroscopy. There's also, you know, RNA folding prediction software, right? So you can kind of
yep.
predict secondary RNA structures, right? So you can determine whether or not what to expect in your product. So like based on, you know, the sequence, you can make a guess as to what it might look like and what you know, some of those 3D structure to, you know, those secondary structures can be predicted. Um, you can also look at, you know, specific enzymatic digestion, right? Where you have ribonucleases that are specific for single-stranded or double-stranded regions, and then sequence those regions to determine whether or not the double-stranded RNA is, you know, due to, you know, secondary structure or it's, you know, matched up with another piece of RNA.
Yep.
So yeah, so there's a lot of different techniques that are out there for analysis that you know in development probably you know I would you know you'd recommend taking a look at and doing evaluation of, especially if you're running into these kinds of issues.
Thanks for that, Adam. I really appreciate at least the insights on the um, separation approaches. So on the second one, can we comment on the high binding of pseudouridynylated RNA to DNA and the ability to remove old DNA?
Yeah.
I think the binding concerns are certainly there, but even right now in most processes, you're not really seeing a ton of binding of the RNA to DNA. It's definitely possible as the RNA polymerase is kind of reading the DNA, and then it's possible for the DNA to loop back and form structures with the translated RNA or just free-floating RNA. But the expectation is that I don't think that binding is very strong. And then on top of that, given that you do perform DNA digestion following IVT, I think the burden is currently low, and certainly process analytics definitely show that. So it's not as big of a concern, at least not based on any kind of published process data that I've seen. And then we were showing the formulation using polymers. We can control the size and encapsulation. Can you comment on uptake and translation of RNA in these polymers? So given that the polymers are biodegradable, as they degrade, they will naturally release RNA. And so, and a lot of times, I think most of the research that I've seen published around polymeric nanoparticles for delivery, uh, the size is really the controlling factor on how it is released. And so, in terms of controlling the size and encapsulation, that has a direct bearing on how much RNA gets released over time. And then some of these polymeric particles, keep in mind, are also autocatalytically degraded. So as they degrade, they'll degrade at a pretty predictable rate. So if you can control the size and you know the degradation rate, then I think the delivery and the release of the RNA is pretty consistent over time.
Yeah. Yeah, and then just the degradation pathway too is important as far as is it diffusional release or is it you know degradation based release, right? So diffusional, obviously the larger particles, the longer it will take. If it's degradation related and you have your if your encapsulation is you know, if it's evenly dispersed throughout the particle, then you can expect more of a linear type of a release. You really got to look at the molecule and how you want it delivered in order to be able to determine what the best approach is to the formulation.
Sure. And then for these last few questions, given that we are over time, would you guys like us to answer these or potentially answer them offline as we've done for past meetings?
Yeah.
Yeah, we'll download this and we'll provide the written answers to the questions.
Okay.
Yeah, let's do that.
Yeah, I think we'll provide written answers to, I guess, all the questions. Is that right?
Yep. Yeah, we can do that.